Dr. Jill Awkerman, who received her PhD in biology from Wake Forest University, where she studied the foraging ecology and population demography of wave albatross in the Galapagos. Since then, she's worked for the Environmental Protection Agency's Gulf Ecology Division, using population modeling to improve risk assessment methods in wildlife ecotoxicology. Some of her projects have included evaluating indirect effects of pesticides on songbird populations, translating toxicology data into ecologically relevant endpoints for amphibian populations, and simulating spatially explicit effects on estuarine fish populations following the Deepwater Horizon oil spill. And that's what she will be speaking about today. So without further ado, Dr. Awkerman.
Thanks very much for having me. As she suggested, I'll be talking a little bit today about how we add ecological relevance to toxicity data. I work at one of EPA's 4 ecology labs, and our focus is on wildlife ecotoxicology. I should start out by saying that this study didn't originate as an evaluation of empirical effects on fish populations. We were looking instead at what we gain from increasing the complexity of different population models in terms of cost benefit and how much we want to invest in decreasing the uncertainty in some of these models. So the approach we take is multifaceted, and it's a population level ecological risk assessment approach. And the reason we're interested in looking at the population level is because some of the effects that you just heard about, physiological, gene expression changes, things like that, may not be lethal, but over time, if they impact enough of the population, could cause declines. And that's what we're most concerned about. So in addition to the laboratory studies that are conducted in very controlled environments under specific conditions, we add the perspective of the field. So do habitat constraints have anything to do with this? Do we need to consider the differential exposure regimes, things like this? We include both of these sources of information into a population model, which provides a very flexible framework for us to simulate different scenarios, look more closely at the range of uncertainty, and see what the potential effects might be. So the model that I'll be talking about today basically has 4 different levels, the most complex models that I'll be showing you. We start with habitat suitability, and this tells us basically where we can expect to find the species within the region that we're concerned. We overlay that with a contaminant distribution layer. So in this case, we're looking at oil distribution throughout Barataria Bay. We put a grid on top of that, and then at each location, we can predict what the population dynamics might look like given the environmental variables and the contaminant concentration. We use the contaminant information and dose response curves that we've constructed based on our laboratory experiments to locally alter the population growth rate according to what we expect at the specific concentration and location. The study species is the sheepshead minnow, not because it's a conservation concern or because it's economically important, but for a lot of the reasons that you heard about the killifish. It's a toxicological model species, so we have a lot of experience in raising this species in the laboratory. It's fairly ubiquitous as well. So along Atlantic and Gulf Coast, you can find this species. It's in our backyard for gathering it, for using in the laboratory, as well as for observing its natural movements and distribution in the Gulf. So what you see on the left is some of our first efforts to translate these organismal effects in the laboratory into field observations of the population dynamics. We know that temperature is important for a lot of these vital rates. For example, fish are going to grow more slowly at colder temperatures, and that's what you see in that first panel. Growth rates dependent on the temperature. The second panel here shows the daily mortality rate for early life stage fish. Fairly consistent, but at the smaller stages, smaller life sizes, they're going to be dependent a little bit more on the temperature. And then that final panel shows embryo survival at a range of different temperatures. And you can see there is an optimal range of embryo survival that is similar to what we find in the summer. And beyond that, survival tends to drop off. So we can use this information to project seasonal fluctuations in our population dynamics throughout the year, looking at conditions in the Gulf. We also incorporated a population limitation of density dependence based on what we typically see in the field. And we divided the life cycle of the sheepshead minnow into 3 distinct life stages. So we had larvae, juveniles, and adults based on size thresholds that we've observed in the lab. We have growth rates, fecundity rates, and survival rates for each stage, and then we can project different effects over time. So like I said, we didn't start out looking to investigate the Deepwater Horizon as a case study, but as we're putting these sheepshead minnow models together, the oil spill happened, and all of a sudden we had a lot of people concerned about where the oil was going and collecting data on the contaminant distribution. So that allowed us the opportunity to include some of these more complex data sources into our model. We're going to be developing spatially explicit population models for the sheepshead minnow, and then using these multiple ecological risk assessment approaches to examine what we gain at different levels of complexity. And the reason we're doing that is because we are looking at broadly applicable concerns in general, so it would pay to have some fairly simple models that were multi-purpose, but to get a really good idea of what's happening, it pays to be as specific as possible and include as much information about the situation as we can. So we want to look at this in terms of cost benefit. Our first layer is our habitat suitability model. So these models were developed using field studies in Pensacola and Choctawhatchee Bays, where we would sample in a variety of different locations and establish the likelihood of sheepshead minnow being present. We collected environmental variable and water quality parameters, and then fit a distribution to those data. When we wanted to transfer this model to predict distribution in Barataria Bay, we looked for the variables that were present for that water body. And we found salinity, dissolved oxygen, and depth were available. So using those 3 parameters alone, we constructed a distribution of where they might be expected to be found. That water body in the bottom right is Barataria Bay in blue. It's got a little red edge around it, which is where we expect to find a sheepshead minnow. This makes sense because they're typically found in shallower environments towards the edge of the water body. And this is how we control movement in appropriate ways of our fish when we're simulating different population dynamics. We then add our contaminant exposure layer. I mentioned that a lot of people were paying attention to where the oil was going. There were these shoreline cleanup and assessment technique observations. These are basically categorical observations of how severe the coastal oiling was. And they were conducted all along impacted areas of the Gulf Coast. So the assumption that I'm using for some of these earlier models is that those visual classifications of coastal oiling are going to represent distinct intervals of PAH exposure within the range that we saw in Barataria Bay. And I'll get back to that assumption at the end of the talk. But we then looked for a 5-year period at what the oiling was within Barataria Bay and used that information, as I described before, to alter our population dynamics and have them projected for the 5-year period. We also collected sediment samples within Barataria Bay for each of the 5 different categories and had those analyzed for total PAH values. Okay, and then using those categories and the estimated PAH values, we adjusted our vital rate parameters accordingly based on experiments that we did in the lab. So we had sediment that most closely resembled the sediment in Barataria Bay based on organic carbon content and particle size. And then we integrated weathered Louisiana sweet crude oil into the sediment at various concentrations. The reason we're focusing on sediment is because sheepshead minnow are found in shallow water. They tend to bury themselves, not only to lay their eggs, but just when they're sort of hiding out in the coastal areas. So we expect that to be a fairly important exposure route for this species. So we did a number of life cycle experiments with them in the laboratory, and you can see some of the same effects that were just described in terms of edema and developmental deformities. But we need to have those presented in vital rates in order to use them in a population model. So That's what you see in the bottom panels here. On the left, you have embryo survival as a function of TPAH concentration. You can see that there's a threshold where it starts dramatically decreasing. The second panel shows our larval stage duration. So as they slowed their developmental rate, it took them longer to reach the juvenile stage. And the same can be said for the juvenile stage. So with increasing TPAH exposure, it took them longer to get to the adult stage. And based on the local estimates of TPAH values, we would alter our vital rates accordingly. You don't have to remember all these, but it just gives you a quick reference into how all these different models are related to each other, how they're similar and how they're different. We'll start off by explaining the 4 spatially explicit population models that we have. The first one represents our baseline or no effects. There's no oil included in that simulation. The second one uses the SCAT categories from the nearest site to determine the estimated TPAH value. And the third includes an oil avoidance behavioral response. So it's been suggested that a lot of the impacts might be mitigated if fish are able to move away from relatively severely oiled areas. So we've included that in our models. They'll move to a cleaner area in the same amount of migration distance. And then finally, we have the homogeneous oil scenario. So here we're recognizing that those spatially explicit contaminant data aren't always available for any given situation. So we take a single location and use that as representative of the entire bay. In this particular case, we use information from Bay Jimmy, which was pretty severely oiled. So this presents arguably a worst case scenario to contrast with our no oil scenario, just to see what sort of results we get comparatively. The next 2 models eliminate the spatial component. So now we have a seasonal matrix model that uses our seasonal fluctuations and the fish population dynamics over time to look at those effects. This allows us the potential to possibly pick up on any population recovery information. And then we have a simple matrix model that just looks at a snapshot in time dependent on the temperature and the oil concentration in that area. We're contrasting all of these with the risk quotient approach. So this is a preliminary screening approach where we go in and we look at the maximum expected concentration and compare it to the lowest NOAC. That's the no observed adverse effect concentration. So what this tells us is that the levels found in Barataria Bay are 65 times that, which are going to cause some sort of adverse effect. But it doesn't give us a lot of context in terms of duration or severity of the impacts. So now I'll go through the results of the models, and we'll start with the more simple models. So on the left, you see the snapshot of the different population growth rates at different levels of oil concentration and different temperatures. The lower temperatures, populations aren't really growing. It's in the summer months at the warmer temperatures where we see the differences in the population growth rate. And the only place where there's an obvious cause for concern is at these more heavily oiled sites. Those are all below 1, which means they're declining populations, but we know that's not representative of the entire bay. So there's some variation in terms of the oil that they're exposed to. The second panel is our seasonal matrix model. So here we compare the no oil in the green line with the oiled site at Bay Jimmy over time, and that's the blue line, the dotted blue line. The red line here is the timing of the Deepwater Horizon spill, and the summer following that event is where we see the most dramatic difference in population growth rates. The summers following that are also different in terms of how quickly the populations are growing, but it's relatively less severe. And then by the time we're 4 years out, these look a lot more similar in terms of the population growth rate between oiled and unoiled populations. Moving on now to the spatially explicit models, I presented all 4 of these scenarios together for comparison. And again, the red line indicates the timing of the spill. And again, we see the no oil in green and the single oiled site, the homogeneous oil in blue, with relatively severe impacts relatively different impacts that first year. We now have two additional scenarios, which is the scat-derived oil contaminant and the oil avoidance, the behavioral response. And there's not a lot of difference in between these two. So the spatial resolution that we were using to simulate the fish movement doesn't really pick up on any sort of differences in how they would move at that level. So not a huge discrepancy there. And again, we see a few years out, these start to get more similar in terms of the abundance of the sheepshead minnow after 4 or 5 years. Now, up until this point, we've been relying on the assumption that these scat categories are meaningful in terms of actual TPAH values to which they're exposed, and that there are distinct regions along that concentration gradient that these are representing. When we look at our sediment concentration values, they don't consistently align up with differences in oiling concentration, in TPAH concentration. And these results were similar to those found by NOAA and other people that had sediment samples analyzed. So when we look at the actual sediment values for PAH, we see a lot of overlap and they tend to be skewed low. We include those in our models. We don't see a lot of difference at all in the sheepshead minnow abundance. A little bit of a difference that first year following the spill, but not much after that. So basically what this tells us is that the visual observations of the oil don't really tell us a lot about the underlying contaminant concentration, which is fine. To be fair, that's not the reason those data were collected. They were collected to facilitate response efforts. But they took a lot of time and energy to collect, and if we had on top of that had to go do analytical chemistry for all of these samples, that would be a huge expense. So this is sort of where we draw the line in terms of what we're willing to invest to have the best possible model and where we can compensate for that with simulations using quantitative data within the realms of possibility of our contaminant distribution. So with that, I'd like to thank a lot of people that contributed to not only the lab research, but the field research as well. And thank you for your time today.